Okay, there you go. So let's start. Years after years, I've been posting this slide for the class, and last time I uh, asked the student to Google it, and when they Google it, it just came up with weird pictures. But of course, I'm talking about a bacteria today, and when a microbiologist will Google, this is what he will come across, and the most dangerous women in US. And that basically is coming from a documentary that came, I believe, 2000 something. And this was coming from the fact, the most dangerous woman in America, coming from the public health hazard. That's where it is coming from, the public health hazard. And then again, it's a very long story. Those of you who are interested can read it up. But for us, today's lecture is interesting because the bacteria that we are going to discuss today has to do something in microbiology history we call, the movie was The Most Dangerous Woman in America, but the actual person was Typhoid Mary. Her name wasn't Typhoid Mary, but she became known as Typhoid Mary. And there were, at that time, there were a lot of articles on that. And she basically was a cook. And she was a carrier of typhoid. And then you can see she's cooking, but you won't see it very clearly. But these are not eggs. These are human head skeletons. So that's what it shows you, that she was considered a public health hazard. So something to do with the public health. I'll just briefly give you her story. Those of you who are interested, her name was Mary Mellon, and she was nicknamed Typhoid Mary. So you can see why do they know her, because she happened to have infected quite a few people, and that caused 3 deaths. And the point that they were trying to make at that time was that they wanted to come up with some guidelines. I'm pretty sure you are familiar with guidelines that you come on any airport and you happen to have an infectious disease or a threat of infectious disease, they quarantine you. And last couple of months, you had some of the people, some of the states denying that. So this is a weird kind of a historical perspective when she was a cook and they kept on chasing her and she would refuse to give her samples because the samples they wanted were the stool samples. So poor girl had to give 200 stool samples over a period of 26 years. Unfortunately, she was quarantined on 2 separate occasions for a total of 26 years. That's weird. 26 years you put a person, and every time she would deny it. But she was posing a threat. So there was a lot of litigation between her and New York Health Department, and they had to fight. But probably the best case scenario was that her own tests were coming negative for obvious reasons. But at that time, probably you can see from here, she was being treated for all the things except anti-typhoid drugs. So probably antibiotics were not considered because she was never found to have that particular bacteria, though she was a carrier. So how did they find out? Unfortunately, they found out at post-mortem when they found out that her gallbladder had Salmonella typhi. So she was passing it on in her feces, and she probably wasn't careful for hand washing, and she loved to become a cook, and she was serving big families in New York, and every time they used to find her, put her into prison, quarantine her, and she would run away. She would change her name, so on and so forth. So that's an interesting movie or interesting chapter if you want to Google it and find out. So the point is that for some of the bacteria especially, if you happen to have as a asymptomatic carrier, you should not be allowed to work in food industry, especially as a cook. So I'm pretty sure FDA, I don't know how much they implement it, but it is there for record for public health that you are not the right person to be there serving other people, because that will be a problem. So today we'll take on a group of gram-negative rods. We'll lump them together, Enterobacteriaceae, as the name suggests, enteric intestine, and they are bacteria, a whole family. And basically they are gram-negative rods. So gram-negative should ring a bell in your mind. And there are tons of that. I will just take on a couple of them today. And the most important you can see is Salmonella, Shigella, and Escherichia coli. So these are the three important gram-negative rods. So if you look at family per se, it has heterogeneous collection of quite a few medically important gram-negative rods, and there are, you can see 40 genera, hundreds of species and subspecies. That makes us think that it's just like a human race. Humanity, everybody is different. Everybody will have a different fingerprint. That's pretty much is happening in bacterial world as well. So if you say Enterobacteriaceae, you probably are talking about those bacteria. They want to adhere, stay, colonize, cause disease, so on and so forth in intestine. We do classify them because we need to treat them. There are quite a few classifications. I'm not going to go in much detail. You can classify them according to their biochemical properties. We can also classify them according to their antigenic structure, DNA hybridization, and then again 16S RNA, ribosomal RNA sequencing, because we used to use antibiotics that are going to target ribosomal RNA. Now, as I said, they are complex, but the good news is that there are only few species that are responsible for human infection. But also keep in mind that this course is mostly talking about human infection, though we do have zoonosis, zoonotic diseases, animals, pets, and so on and so forth. And then now and then you hear in the news that poultry, eggs, and so on and so forth contaminated with salmonella. So there's always something going on with the regulations that we have. They are present everywhere, as you can see: soil, water, vegetation. They are also normal intestinal flora of most animals and humans. Okay? And then again, remember, I'm just talking about the whole family. I'm not talking about one specific particular pathogenic bacteria, because I just told you, even if you have that as a normal flora, if you are a carrier, then you may not pose a danger to yourself, but you do pose a danger if you happen to work, as I said, food industries. The most important thing for Enterobacteriaceae is that they are responsible for about 30 to 35% for all bacteremias that we see. And then again, 70% of urinary tract infection. So these are some of the higher incidence of diseases that we normally see. UTI, 70% is a lot. And they are Enterobacteriaceae. And then, of course, intestinal infections, because that's where they sit and reside. Now, as I said earlier, some of these organisms are always associated with human disease. They are called pathogenic, virulent. So if you find them, basically, they are going to cause disease. They are Salmonella, serotype typhi, Shigella, and Yersinia pestis. What does Yersinia pestis cause? Plague. That's also Enterobacteriaceae. And then some of them, like E. coli, Klebsiella, and Proteus mirabilis, they are also member of normal commensal, but again, they're opportunistic bacteria. So it suggests to you that if you give them an opportunity, if there's a cut, if your immunity goes down, and anything that compromises you, they will get an opportunity to cause infection. And then finally, we have third group, which are again normal commensals, and they themselves by nature are not pathogenic, but of course they can acquire virulence factor genes. So again, plasmids, are the one that will bring those kind of factors into incorporate them into so non-pathogenic bacteria. And typical example is E. coli. So E. coli is a typical example that we normally see. Now, most of the time, if you want to look at where is the reservoir of these bacteria, because they are in human intestine, they are in animal intestine, so any people, any person, even ourselves, we are meat consumers, dairy product users, so on and so forth. So one way or the other, we will come in contact with them. So Salmonella species and Yersinia, they are coming from animal reservoir. Human carriers, I just told you, especially for the Typhoid Mary, Salmonella serotype typhi, and Shigella. And then again, we may have endogenous spread of organism in susceptible patient. Endogenous mean that your own normal flora is coming and causing a problem as compared to exogenous, where you're relying on another source. And then again, E. coli basically is the major culprit, Escherichia coli, that probably can infect and invade almost all our body sites. So if you look at statistics, you will see especially the whole family of Enterobacteriaceae in order of prevalence. So if I was to ask you, this is one of the commonest questions asked, that if I was to ask you, let's take an example for lower respiratory tract, which of the following three has a highest prevalence of infection? So the list says Klebsiella, Enterobacter, and Escherichia coli. So the way the list is made, number one has a highest prevalence. So highest prevalence for bloodstream infection, Escherichia, highest prevalence for gastrointestinal tract, Salmonella, highest prevalence for urinary tract, Escherichia again, and central nervous system as well. So that's why we talk of prevalences and incidences, but these things can change. There are statistics maintained by the public health authorities, by CDC, by WHO, and so forth, that may change over time. But for reference purposes, for your test purposes, for your exam purposes, I would take whatever your book says as a normal and current as of today. But this is how they maintain. So if you look at incidence of Enterobacteriaceae associated with bacteremia, because bacteremia is when you have sepsis, you have bacteria running in your blood. So wherever the blood takes is going to cause infection. So you can see from here, 45% of the time, bacteremia, Escherichia coli, 22% Klebsiella, 20% Enterobacter, and so on and so forth. So the question usually will be asked, which of the following have the highest prevalence of causing bacteremia? Then of course the answers are Escherichia coli, Klebsiella, Enterobacter, whereas the other Proteus, Serratia, and Citrobacters can also cause disease. So this is what, especially when it comes to public health, when it comes to incidences and prevalences, that's what we want you to know about epidemiology of some of the infections that are there. Gram staining, so you can see Salmonella, positive blood culture, and this is a typical, we call it bipolar staining, because if you look at the rod, both the distal ends will have a higher staining. So you'll see the distal ends on the sides more stained than the middle. So that is called a bipolar staining. Okay? So in the next slide, of course, we'll talk about pathogenesis. So we're just going again in the same sequence. We talk about the physiology and structure. We talk about the journal family. We talk of some of the, right now we're just going through the journal family, that what are the journal characteristics of most of the Enterobacteriaceae. So if you look at the virulence factor that we discussed in the last lecture, again, being an gram negative, they have an endotoxin. They have a capsule. They have an antigenic phase variation. They have type 3 secretory system. They have tons of growth factors, and then they are resistant to serum killing, and they have antimicrobial resistance, and so on and so forth. So you can see, the more there is a list for the virulence factor, the more problem that we will come across in terms of having disease. Now, if you look at the structure of A Enterobacteriaceae, remember, again, you will see the most important thing that I want you to pay attention is a lipopolysaccharide or endotoxin. And you can see it has a, from outside in O polysaccharide. Then middle we have polysaccharide core, common antigen, and the actual endotoxin is lipid A. So this whole structure is called lipopolysaccharide because it has a lipo portion and a polysaccharide portion. O is an external most, and lipid A is the real toxin, and you can see this embedded in the outer surface of this bacteria. Then again, we have a typical cytoplasm. We have inner membrane. We have peptidoglycan, small lane, and then we have a capsule on the external end, which has K and VI antigen. And then again, you pretty much are familiar with a typical gram-negative cell wall where you have a periplasmic space as well. And that's where quite a few those enzymes like beta-lactamases are sitting. And that's where we have a problem of using antibiotics. The other important thing you will see in the structure over here is that they have flagella, which is an antigen. So flagella will give these bacteria additional motility, additional kind of a push to invade and push around and go into territories that normally other bacteria we don't have flagella would not be able to do. So the most important thing, as I said in this one, is basically the structure of a lipopolysaccharide, and also make sure, and you can make a note on your own, what is the difference between lipopolysaccharide and lipooligosaccharide. So these are the two important things that sometimes is asked in the exam. If you look at generally, they are gram-negative, facultative anaerobic rods. And why they are anaerobic? Because they tend to sit in intestine and there's no obvious source of air there. And they ferment, so they ferment sugar. So we do have, I'm pretty sure those of you who want to go in detail, we have lactose fermenters and lactose non-fermenters and so on and so forth. So their ability to ferment a particular glucose molecule can be used as a means of classification. And then again, you can see 2,500 O serotypes. They are tolerant of acid and phagocytic vesicles. So that's again very important thing, because this means if you phagocytose them, they're not going to be killed because they can survive. So they are also a part of the intracellular survival cells.
So they don't contain protein A, they just are able to survive the acids of the lysosome?
What do you mean protein A?
Aren't those bacteria that avoid phagocytosis, don't they avoid it because of protein A?
Not necessarily. Protein A basically is different in different bacteria. For example, in this one, we had it as a part of LPS. Some of the bacteria may have different proteins. But if you're interested in exactly the mechanism, we'll talk about that. Yeah, but every bacteria may have a different mechanism to survive in macrophages and avoid phagocytic vesicles and be tolerant to acids as well, because it has to pass through your stomach. So you have to eat a food contaminated with salmonella. So of course, your stomach acid is not killing them. Otherwise, they would have been done away with. And then again, you can see the diseases that cause inflammation of intestine. They call enteric fever, which also called typhoid fever. They call bacteremia, we already saw that. And then again, as I said earlier, as the impact slide, you can be an asymptomatic carrier like the Typhoid Mary case. If you look at epidemiology, you will see that most of the infections are coming from eating contaminated food. So poultry products and other sources of infection are important. As I said, they are enteric, so people who are not mindful of washing their hands. So fecal-oral route is common, especially in children. And also keep in mind that Salmonella typhi and paratyphi are strict human pathogens. This means they have no home other than humans. There's no other reservoir for them. And then again, they can be passed from person to person. And as in the case of the Typhoid Mary, you saw that she herself was okay, because that's what she used to claim, I'm okay. Well, she was okay, but she was passing on her infection to others. The question you may ask is that who is at risk? So of course, those people who eat improperly cooked poultry or eggs, or patients with reduced gastric acid levels, because the thing is this, that if you reduce gastric levels, you basically will allow quite a few other bacteria. That's why I said the chronic use of antacid is not good, because you are giving a chance to bacteria to pass that barrier of stomach and go. Because these, when we say enteric, we're talking about small intestine. So they've already passed through that. And then again, if you're immunocompromised, because when I say these is a bacteria for intracellular survival, what I mean is a T-cell mediated immunity. And if you're immunocompromised, your T-cell mediated immunity is lacking, so you may have a chance. And infections are prevalent all around the year, especially in warm months all over the world. And the diagnosis basically is, as I said, a stool specimen. So it's a stool specimen where they are looking for an antigen, looking for bacteria to diagnose you. If you have a mild enteritis, for example, Salmonella enteritis, so you let your immune system take care of it. We don't give antibiotic treatment. But if you have infection with Salmonella typhi or a paratyphi where it gets disseminated, so we will rather treat you with the drugs that we have. Fluoroquinolones are important. Chloramphenicol used to be a drug of choice, but again, it has quite a few strong side effects. One of the side effects of that is that it affects the bone marrow. Trimethoprim, sulfamethoxazole, or broad-spectrum cephalosporin. I know that you have not started learning them, but for typhoid, they're simple drugs. They're simple drugs. They are called the drugs, antibiotics for typhoid. And then again, for public health awareness, most infection can be controlled. FDA does regulate that. You want to make sure that the eggs that you get, the milk that you get, the meat that you get is not contaminated from those bacteria. And carriers for Salmonella typhi and paratyphi exist. They should be treated as well. Vaccination is available for travelers. If you were to travel, Asia and Far East are the hotbeds of the infection, in Africa as well. So the WHO report is their water is unfit for human consumption. So you want to make sure that you get vaccinated if you were to travel to all those countries. And again, Department of State does run all these kind of websites to aware you of what is going on and what is it that you need in terms of vaccination. We already discussed that. In US, mostly is gastroenteritis, but septicemia is also there. And the prototype, the classical kind of infection that you get, in that part of the world is typhoid. And I must have said it many times. One of my professors in England used to say that if you want to see a disease in a very early stage, probably you have to see in the West. But if you want to see a disease in a very advanced stage, you either have to go to East or Africa, because people will keep on doing what they're doing till it becomes to such a level that their intestines will perforate and they have peritonitis because of typhoid, because of many problems that are out there. But we basically in this country will have a mild salmonellosis, gastroenteritis. People may have food poisoning, and then we can get it over with. If you look at the historical pathogenic picture, so you can see that both Salmonella and Shigella, I usually consider them like a twin sisters, because many things are common. Because they are all foodborne diseases. And most of it you can see for animals, for example, on the right-hand side, if your source of food is one way or the other coming from animals, Salmonella and E. coli are the one that should be suspected. But if it's not directly animal, but it can be food, and again, those people who don't wash their hands after using washroom and public lavatories and so on and so forth. So you basically inhale. So you see fecal oral. So that's what the transmission is. So people will have that either pass out from the system. And again, why the incidence of UTI is high? Because, especially in the female, because of the close proximity of a perineal area where you have an anal canal and the urogenital canal very close, and then the bacteria can migrate up into urinary bladder and all the way to the kidneys. And again, you can see that sometime you can also get a cut, especially for Shigella side, but most of the time, as I said earlier, bacteremia is where you have actual bacteria running in your blood. And this is a kind of an upshot of what may happen in terms of getting pathogenesis. So I got this kind of classical picture from one of the books, but they should be there. So let me complete the lecture and I'll take your question afterwards. Yeah. Sorry? Well, I said that all the slides which are presented as a podcast are loaded on the Moodle, or you have an option to download PowerPoint slide as well as listen to them. Okay?
No, but I'm saying that the previous slide, we don't have that.
That's what I'm answering. All the slides are posted on the Moodle. You just need to download them from there, right? Okay, because I keep on adding slide till I present. Even this morning I was looking at CDC data. I keep on changing that. Okay? The question you may ask is that, what is it that you need to know from there? That's what the point is. Okay. So you can see from here again, a perspective that I want to give it to you, especially because the theme is public health awareness. So for you just to know that typhoid causes salmonella, typhoid causes typhoid is not enough. That information is not enough. So for you, you need to look at the incidences, prevalences, and we have to deal with the local incidences, your state level, hospital level, because things are different. So you can see from here that if a person, for example, having, let's say, typhoid, what are the chances that he's going to have fever? And what is it that a person who presents with that Enterobacteriaceae infection and he has an pneumonia as well? So we are looking at the percentages, and you can see that when we talk of those generic symptoms, everybody does not fit into that. So we are looking at percentages. For example, most of the time, epidemiological questions are such, like for example, if I was to ask you, do the patient suffering from typhoid will always have enlarged liver and enlarged spleen? The answer is no, because only 15 to 35% will have it. But we do put it as one of the sequences. Now again, the question will be that when you even run for the blood test, you may not get everybody positive. Remember we talked about Neisseria gonorrhea, and that young 17-year-old girl had all her tests negative. The only test that came positive when they did a pap smear from her cervix. So don't get misled by that. Sometimes we are unable to grow. Sometimes there are many reasons for us to miss that. So you can see that blood tests basically are the culture test. And those of you who have never seen people going for the culture, they would take 20 ml of blood from your right arm, 20 ml from your left arm, right? And then do all this testing. You need a lot of volume of blood to do testing because bacteria can disappear. So you can see from here, people may have presentation in anemia. They may have sometime, if a person has a problem, because especially as I just said, you must have heard me saying that, well, typhoid is not an issue. So you only need to have to care for you getting typhoid when you travel to Asia or travel to Africa. But there's always an exception. And the exception is if you have HIV. If you have HIV, your T cells are compromised. You need T cells to eliminate this bacteria. So AIDS patients have given a new perspective, especially for the Western world, to explore and find out what is going on in terms of these infectious diseases. So these are infectious diseases. The biggest challenge that we are going to get in medicine is current infectious diseases. There are new diseases coming up. That's why I myself have a special elective for current infectious diseases, because they keep on popping up every year. Sometimes they're bacterial, some they are viral. So you have to keep in mind that when I say gastroenteritis, for example, diarrhea may not be the prominent feature. But sometimes when you memorize things and you say 5 things, you tend to think that it has to have a diarrhea, it has to have hepatosplenomegaly, it has to have disseminated infections, so on and so forth. So keep your mind open in terms of incidences and prevalences. That's what the whole point is. Now also you can see from here, if you look at some of the statistics that we run, especially because right now we teach you the human pathology. We teach you some of the things which are affecting human as well. But also, as I said, we have a lateral link with animals as well because of consumption of meat and dairy products. So you can see from here is going to respond. And we divide that into a issue in terms of you having a very, very mild infection. You can see on the left-hand side, just a diarrheal disease, to a very invasive and a systemic disease. So obviously, it goes along with the mortality data that if you have a mild disease, low mortality. And if you have an invasive disease, you have a high mortality. And many times what we do is that we see some of the animals, for example, in this case, the major meat is coming from pig and cows, then again, they pretty much have got adapted to these bacteria. Because I may not have told you earlier, the biggest consumption of antibiotics is not humans. The biggest consumption of antibiotics is animals. That's again a big lobby over there, because you say we don't give antibiotics to the patients, we don't take it ourselves. But the food, the meat that you're eating is already been treated with antibiotics because either animals were treated with that or foods were treated with that. So that's the basic consumption. I want to say, put a number, but say 80-90% of the stock of antibiotic is going towards that. So that will explain to you the discrepancy that we may have in our system. Okay? All right. Now, also keep in mind that since the technology is changing, there was a time for us to just look at for the culture, but now we can do even a genome. So these are genomic studies. So we basically, as we have mapped up a human genome, we have also mapped up bacterial genome as well. So we can pretty much find out what particular type, genotype, you can see, or a phenotype is particularly important in animals, humans, birds, and so on and so forth. Again, if you look at the immune response, this is a few slides for immune response, just to recap, because I keep on visiting it over and again. I think your book just talks about an immune response. So you need to have CD4 T cells, because CD4 T cells are required. And when I say CD4 T cell, the best scenario for you to remember is a person, if he has a HIV, and then normally how many CD4 T cells you have for every one CD8 T cell? 2 is to 1, correct. So the ratio is reversed. So we need, you can see from here, that if you do not have a patent T cell immunity, you are prone to quite a few infections, especially Salmonella infections. So Salmonella, as I said earlier, is very obvious. So you'll just have a spot, you can see from here on an intestine, and that's where it will grow and then pass out into your system. If you want to look at the closer look, how it passes into the system, you can see bacterium over here, especially in gallbladder. People do get gallbladder infections, and sometimes this bacteria sits in gallbladder for a long time, and then they can kind of wrap themselves around the gallstones as well. So those of you who are interested, they find out that one of the commonest cause of gallbladder infection is also Enterobacteriaceae, especially Salmonella. And their ability to basically cause damage as well. Because remember, as I said the other day when I was talking about the pathogenesis factor, some of the bacteria will invite neutrophils. And these neutrophils are not being invited to help them, but to cause a disease. So you can see from here, they release those chemokines, and we have already discussed that. So gallbladder, you can see the stone of gallbladder on the top. And then these stones basically are a very nice kind of a mesh for this salmonella to hang around and be a part of it. And then you can see when the stones are going to go and pass out through the common bile tract into duodenum, they will also bring salmonella as well. So many times people who have acute gallbladder infections, we do see that salmonella is an important aspect. Okay, and again, there are some good cases in your book for those of you who want to correlate that with the clinical aspect, because I always think that would help. And you can see from here, you look at a 25-year-old woman. She's coming from Philippines. She has enlarged liver, and she is tested positive for S. typhi. So again, typical story. When you see a patient, we have both liver enlarged, spleen enlarged, and high fever, and then coming from an area where it is highly endemic, is always there. Okay, Shigella basically again replicates in colon. That's the only difference as compared to the previous one. As I said, they're like twin sisters. If you look at the structure, pretty much the same. The only difference is that they have 4 species, and all 4 are important, but especially, I mean, it's like a dichotomy, and I don't know, I can't give you a reason for that, that Shigella sonnei is responsible for infection in the West, and Shigella flexneri is there in underdeveloped countries. And then again, if you look at very severe infections, especially diarrhea, Shigella bodii is commonly isolated. So these are some of the important things. The other difference between Shigella and Salmonella is that it doesn't come with endotoxin alone, it comes with exotoxin as well. So you can see Shiga toxin that causes damage. And many a time, mild form of gastroenteritis we call shigellosis. And sometimes, remember as a rule, when the bacteria touches the superficial surface of intestine, it irritates and causes diarrhea, just basically watery diarrhea. But it invades and goes into submucosa, that's where the vessels are, we got bloody diarrhea. So if you saw in the previous picture, the more invasive the bacteria is, the more blood will come out. So we have a bloody diarrhea and bloody stools. And dysentery, basically bacterial dysentery, is important, and sometimes people may or may not have fever. If you look at epidemiology, only humans are reservoir, fecal oral route. Patients at higher risk are those people who cannot wash their hands, are too small, too old, or don't care. But one special thing for this one is that this is also known to be very common, especially Shigella, in male homosexuals. So that's one thing that you will see a difference. As I've been saying to you, that if you want to use those 5 objectives horizontally across the different bacteria, some of the things which are different are usually some of the things that are usually asked. Another important thing for this one is that you just need few organisms to produce disease. And if you only need few organisms, as I said the other day, 1 organism for TB, so the word that we normally use in medicine is that this bacteria is highly infectious as compared to Vibrio cholera, where you need tons of them. So you can see highly infection. Again, depends upon where the disease is. And again, many times if the inoculum means that you get exposed to a very small, and you see today's lab as well, when you take that loop and you take that, burn it up and take that swab, so that is inoculum. So whatever comes in that loop is called inoculum, bacterial inoculum. So you can see if it's a low inoculum, many of you didn't get gram staining because the bacterial concentration in that inoculum was less. So that's why you didn't grow them on gram staining. But you cannot tell. So if you get exposed to a low inoculum, probably you may not get a disease. But again, depends upon many other factors as well. But we do want to test your stool to find out if you're really suffering from that. As far as treatment is concerned, of course, for asymptomatic, we will not do anything. The other thing that you have to keep in mind for Shigella is that there is a fecal shedding. And of course, any bacteria that has a route of spread fecal oral, this means that is passed through feces. So it lines the intestine, so you shed off in feces, and if you don't wash your hands, then you'll get it. We start therapy ASAP with fluoroquinolones or trimethoprim, and then again, you have to make sure that the proper hygiene is taken care of when you are seeing the patient, or you happen to be a nurse and you come across anybody suffering in your household. Hand washing is an important thing. And the clinical disease, we already talked about that, is as Salmonellosis, shigellosis, pretty much the same. So the last bacteria is Escherichia coli, and that's the one that you normally see every day. I mean, I'm pretty sure if you even say 2015, you say Escherichia coli, there always are multi-state outbreaks. And either your spinach is contaminated or something is happening. Something is always happening with Escherichia coli, and these are gram-negative facultative anaerobes. They are oxidase negative, they have a lipopolysaccharide, and they have 5 different pathogenic groups, and I want you to remember all the 5. And they are the most important one, and I'm going to tell you how to remember it. They may look difficult, but they're very, very easy to remember. EPEC, ETEC, AHEC, EIEC, and EAEC. Pretty much the last 2 words are Escherichia coli. That's a common thing. The first 2, they represent pathogenesis, how invasive they are. And especially, why does the government or healthcare are so much worried? Because it causes a very fatal problem, hemolytic uremic syndrome in this country. And then again, it can also cause extraintestinal disease like meningitis, UTI, and intra-abdominal infection. And I would recommend you that you go and try to kind of memorize or whatever you want to do with this beautiful picture that will come, because in clinical practice, 70% of your cases are E. coli. 70% of your cases urinary tract infection. So site of action, you can see the top 3 are small intestine, and the site of action of the last 2 are the large intestine. Now they cause a disease, but I want you to pay attention to the pathogenesis as well, because once you understand the pathogenesis, it's very easy for you to understand the disease. Now, enterotoxigenic, ET, so you will see basically it will have a mild kind of a picture because it is just releasing a toxin. And this toxin basically is coming to this E. coli from another source, which is plasmid mediated. If you pay attention to enteropathogenic, which is a little bit more severe than that, again, you will see a mild kind of diarrhea, because when I say mild, I mean no blood, no blood, no blood. Again, it's a plasmid-mediated infection, and then again, it will disrupt the microvillus structure, a little bit of damage, as compared to this one, which will just cause hypersecretion, and we'll call malabsorption. Now, if you move up, you will see enteroaggregative, and if you remember, I said that the aggregation substance is one of the virulence factor. And again, you will see a adherence of that plasmid mediator. So 1, 2, 3 are plasmid mediators. And this way you cause you basically have shortening of microvilli, so the anatomy of the intestine is changed, and you have hemorrhage. So now you begin to see the disease over here. And then last two basically are in large intestine, where the real problem is because you have a hemorrhagic colitis, very severe form of colic, bloody diarrhea, and then again causing hemolytic uremic syndrome, especially AHEC. And you will see it typically by cytotoxin, Shiga toxin 1 and 2. So this basically is the one that stands out because it causes one of the major problems that we see in this country, and all others basically are considered as the mild form. So if you Google and find out, you will see that pattern over there. Very common, aerobic, most infections are endogenous. This means that they're coming from you. If you want to look at the diagnosis, you have to do a culture, or the most important thing is, with the exception of AHEC, they basically are only detected in reference, because they don't do it over here. They will only do it because on the reference lab by the government. How do you treat them? Again, depends upon the symptom. Before you initiate antibiotic therapy, you want to make sure that you do in vitro susceptibility tests, which many of you will do today. You put that disc on the bacteria, and then again, you want to make sure that you have appropriate infection control, especially in the hospital settings, and then maintain very high standards for, especially if you're going to reduce the gastrointestinal stain. And then again, also proper cooking is what is most important. And I'm not going to go through some of the other slides, but they are there for you to upload. One of the slides is there important in terms of the difference that E. coli enterotoxin have. So you can see one causes elevation of CMP, the other was basically cause elevation of CGMP, because these are important in terms, remember I said to you that we are making drugs learning from how this bacteria will cause a disease. So if you have elevation of cyclic AMP, you lose the secretions. So you lose the secretion, that's why you lose electrolytes, and you lose water. So these are some of the important things that we normally see. And again, for especially this group, for Enterobacteriaceae, I would suggest to you that you can compare these three important ones, and also the table that I just mentioned, and spend some minutes on that. So you should be aware as to what kind of pathogenesis these bacteria are causing. And then also, which I didn't discuss at that time, keep in mind that when we talk of this mild thing, it's usually either small children or infants involved, or people having a mild symptoms like traveler's diarrhea. And then, but the most severe form, we see that in senior citizens and people who tend to be a little bit of disabled in terms of helping them out, washing their hands and taking care for themselves hygienically. Okay? And there is one case, I think, in the end, and you can also see on your case, on your website for the book. I was told that especially for the flashcard and the case studies are very good on the website of Morris textbook. And then again, you will see a typical inflammatory disease. The only differential diagnosis is that these are infectious diarrhea. These are not diarrhea because of chronic inflammatory disease like Crohn's disease and ulcerative colitis. So these are not the two. So you have to exclude those ones before you register a case for these ones. Okay? So I'll stop here, and thanks.